Okay, so thank you very much. So, Just has been talking about obtaining mitochondria from a population of cells that's present within the ovary, and I'm going to talk to you now about the isolation and purification of this cell type. Now, these are a cell type that's had a lot of controversy, and there's many groups that have been trying to isolate them, and there's many groups that can't isolate them. So that has led to an increase in the controversy. So every time something's published about these cells, there's another paper that comes and says, well, these cells, we can't get them, therefore they don't exist. So I'm going to tell you how I came to work on these cells and the type of cells that we're isolating in Edinburgh. So I'll just acknowledge my funding. The funding for the work that I'm going to talk to you about is from the Medical Research Council. But we now have funding from Ovascience, but it's not related to, it's related, but it's not the work that I'm going to talk to you about today. Okay, so most of you are aware of this, and this is the idea that we have, and we've had for a number of years, that the population of primordial follicles is laid down before birth in humans. And that doesn't change. Of course it is. There's a population of primordial follicles that are laid down before birth in humans. Nothing changes in our concept of that. And we know that these follicles are initiated to grow, and many of them are lost, and in a monoovular species, only one will ovulate. So there's a lot of loss and degeneration of oocytes. But the question is whether the ovary has any capacity for regeneration. Now, if I was giving this talk in the 1920s, we would actually be talking about regeneration, because that was the prevalent scientific idea that the ovary was capable of regeneration. But since the work of Solly Zuckerman, everyone has accepted that there's no regeneration within the mammalian ovary. Now, why has this re-emerged? Of course, it's a very hot topic, and it has been for a few years now. And that's because of the emergence of the field of adult stem cells. So again, a few years ago, we wouldn't have accepted that there were stem cells in many of these organ systems. And we now know, because of this work at the adult stem cell field, that there are stem cells in almost every organ system for every tissue that's been looked at. So it makes sense that the ovary would then have stem cells residing within the mature ovary, particularly for the somatic cell component. But why would it not have cells that could perhaps contribute to germline? And that's what many people have been looking at. And the group that was probably the main group working in this field was a group of Jonathan Tilley, who did a lot of work on the mouse, and the Chinese groups of Jiu Wu, who worked on the mouse as well. But in 2012, the group of Tilley had a breakthrough in that they could isolate a population of cells from the human ovarian cortex. And what they found was that they had these cells that they could identify by DDX4, which is the same as VASA. It's a germ cell marker. In the ovary, it's always been associated with the germline. But DDX4 exists in other tissues. It's highly expressed in the brain, it's in the lung, and it's in the kidney. It's in many tissues. The only tissue that we found that it doesn't express is in the skeletal muscle. But within the ovary, it was only ever found in the germline. So they pulled down these cells using DDX4, using a DDX4 antibody, and they found that these cells had germline characteristics, but could also proliferate. And of course, a oocyte doesn't proliferate, so they led to the conclusion that these were some kind of germline stem cells. Now, this is just a table giving a definition of the types of stem cells. And this type of cell that Tilley was characterizing was a unipotent cell. So a cell that had a particular lineage, that it was going to go down the germline lineage. And that's what he was working on and trying to get them to develop to become an oocyte. So when they isolated these cells, they isolated them from human adult women the ovarian cortex, and they used an antibody to DDX4 to pull down these cells using a FACS sorting technique. And they also then transfected the cells with the green fluorescent protein so they could trace the cells. And they then took those cells and injected them into a piece of human ovarian cortex again, and then transplanted it under the kidney capsule of the mouse, the immunodeficient mouse, and then they looked at the histology after between 7 and 14 days in the kidney capsule. And what they found was that they had oocytes within follicles that were green. So those oocytes had to have come from the cells that were injected. And they looked like oocytes, so the conclusion was that these cells were indeed a cell type that could go down the germline pathway of differentiation. So at that time, I was incredibly sceptical of these cells, and I know many people were very sceptical as well. And there was a lot of discussions, a lot of debate, and there was a lot of comments that were published at that time. And then Jonathan Tilley approached me, and he asked if I would test his cells and see what the potential of those cells were in vitro. And the reason that he approached my group was because we had been working on, and we continue to work on, an in vitro system to grow immature human oocytes. So what we do is we take the human ovarian cortex, we prepare this cortex, place it in vitro, and we get activation of the primordial follicles. They start to grow in vitro to a stage where we can dissect them out when they're preantral follicles, and then we can grow them to the antral stage. We can remove the oocyte and its surrounding cells, and we can then mature them further or grow them further, and then we can mature them, and a proportion of these oocytes can reach metaphase 2. So we had a system where it seemed that we had the potential to test the ability of these cells to really form oocytes, and that's what we did. And of course, when the cells came to our lab, we were incredibly sceptical. We never for a minute thought that this was going to do anything. And what we did was we took the cells that were labelled with the green fluorescent protein, we injected them into the ovarian cortex, and then we monitored them. And this is what they looked like after 7 days. If we took the tissue at 7 days and prepared the histology, we could see small green cells. And these would be the cells that were injected into our pieces of tissue. But then we could see larger structures. And of course, we didn't have enough definition to really know what they were. So we then looked at them by classical histology using a chromogenic marker for DDX4. And what we could find when we monitored the cells that way, we could see what we would define as 4 types of cells. So here's the cells that we would say are the cells that we injected in. They're small cells, GFP positive. And then we could see what we would refer to as type 2 cells, where the cell's enlarging and it's maybe starting to try and recruit somatic cells. Type 3 is recruiting somatic cells. And type 4 is, I'm sure anyone who's looked at ovarian histology would say, it looks like a primordial follicle. And that's indeed what it looks like. And of course, we were very sceptical, and we did all the sort of tests to see, does GFP link into the oocyte or not? And, you know, that was just ridiculous. It had to have come from the cells that we injected in. The only place you saw GFP was in certain oocytes. So here you see, side by side, an oocyte without GFP, and that's an oocyte that was already in the ovarian cortex, and then an oocyte that's GFP positive, and that's one that has to have come from the cells that we injected in. And maybe we might not call them oocytes, we might call them putative oocytes, because of course you have to test whether they're real oocytes, and the only test for that is to see if they can be fertilized and form embryos, and if they form live offspring. And of course, this is human material, so we're limited in how far we can actually take that testing. But this summarises these experiments where we had our culture system, We injected the cells into the tissue, and then we could see the development of oocytes that were derived from the cells, and these could form multilaminar structures. So that was quite a shock to us, but it was a really interesting finding. And we then became exceptionally curious about the cells that were in the ovary. And we know that the ovary does have so much activity and that there are many, many other types of stem cells. So for us, it became important to see whether we could isolate this particular cell that could go down the germline. And that's what we started doing in the lab, and that's the people that were involved in the work. So we, instead of taking cells from another lab, we wanted to see how easy or how reproducible this could be. And it's not easy. It's difficult. And it's difficult because the antibody that's being used is a DDX4 antibody. And the ovary is full of DDX4 when you mash up that tissue, because there's DDX4 in all of those oocytes. So you have to really discriminate between what's a real binding and what's just non-specific binding because of the DDX4 that's going to be sticking to other cells. So there's a lot of work that's required in terms of the isolation procedure and also in the FACS-based sorting. It's not a case of just throwing it in and getting it out. A lot of groundwork has to be done. And we had really talented people working in our lab. A talented clinical research fellow, Sherilyn Lopp, worked on these cells, and a postdoc, Yvonne Clarkson, another postdoc, Marie McLaughlin, have been working hard to work out the best way to isolate the population of cells. And they've done a lot of work in this, but essentially it is using DDX4 as the antibody to pull them down. But we also use other markers, and we also use size as a discriminator. So we're looking to see a particular size of cell that we can isolate from the FACS-based protocol. So this just illustrates what the FACS system is. I won't go into too much detail on that. But what we found is, because of the FACS sorting, we decided that we would look at extreme parts of our FACS sort. And we believed that there was several populations of cells. It wasn't just a homogeneous population that would be sorted out. It was different populations. And that might explain some of the differences in results from various labs, because you could be selecting for a particular cell type. And we did that. We sort of looked at different areas of our FACS plot, and then we did the molecular analysis on these. And what we found was that, yes, that it appears that there are subpopulations. And this is something that we need to look at in detail, because these could be cells that are at different stages of differentiation. And of course, that would make sense if you have a cell that can go down a particular pathway. You wouldn't expect necessarily that you would get a homogeneous population. So we found populations that had germline markers, stem cell markers, no follicular oocyte markers, so they're not oocytes, and what we found was that there may well be splice variants present in some of these populations of cells, but in one population we could see dazzle quite clearly. In some of the populations we didn't see dazzle, so we don't know if that represents a different stage of differentiation, and that's what we're hoping to look at in more detail. But there's been a lot in the literature that says, well, you're using a DDX4 antibody, but other people, when they've been working in mouse, no one has said this in human, but in mouse, they've not been able to isolate they can isolate cells using DDX4 antibody, but the cells don't express DDX4. And that's not our experience at all. Our experience with the positive cells, they express DDX4 at the messenger RNA level and at the protein level. So when we've done the blots using different antibodies, we always see the protein in the freshly isolated cells. We see it positive for DDX4. And as I say, we've used two different antibodies. We don't see it in skeletal muscle. That's about the only tissue that doesn't appear to have any DDX4, so it's a good negative control. But when we culture these cells, sometimes we find cultured cells that don't express DDX4. And that might be that there are certain populations that are, being grown out, or they're taken over, and we're not seeing the specific ones. But the freshly isolated ones do have DDX4. So the question then is, if these cells are there in the ovary, using the DDX4 antibody, we should be able to see them in tissue of ovarian cortex. And if you look here, this is a human ovarian cortex, stained positive, the oocyte stained positive for DDX4. And I don't think anyone would argue that it's not DDX4 because it's been an accepted marker of the oocyte for many, many years. But when we go through the tissue, we find, and this is laborious, you have to go through every section and look for these cells, we find small cells that are positive for DDX4. So we would suggest that those are the cells that, in fact, we're pulling down and are growing, that look as if they're growing towards oocyte development. Now, just to address that further, because again, this is a big controversy about the whole thing about DDX4, and the people working on the mouse seem to have issues with DDX4 actually being there. Now, DDX4, if it's being pulling down cells, it should be in the surface of the cell. And there's some papers, publications that say the DDX4 cannot be expressed on the surface, that it's an RNA helicase and it's not expressed on the surface. Well, there's no hard evidence for that at all. But if you're pulling down using a DDX4 antibody, that would suggest that they must be in the surface. So what we've done is we've designed, we've been able to insert a full-length DDX4 into cells, and we've put a tag at the C-terminus of our DDX4. And what we found is, like, if we take that tag, so it's a fluorescent tag, and this is one that it would be internal, this is the control. If you then stain it up in non-permeabilized cells, if it's not on the surface, then you won't see it in non-permeabilized cells. So when the DDX4 that we've inserted in, we do see it on the surface. So it can be expressed on the surface under these experimental conditions. So we are fairly confident that what we're pulling down is actually DDX4 positive cells in the human. The mouse may be different, but certainly that's what we are seeing in the human. So just to finish off quickly so that Bob can talk to you about the clinical work, then of course with these cells that we're isolating in the lab, we're testing the potential. And we're doing a lot of in vitro work with the human cells. But I just want to show you some of the work that we've done with our bovine cells. We've also isolated these cells from the cow, and we've done these chimeric ovary experiments where we've taken these cells, the putative oogonial stem cells, we've combined them with mouse somatic cells, so there can be no argument about where these cells come from if we can identify bovine cells or mouse cells. And we've then put them under the kidney capsule of the SCID mouse. And this is the work of a PhD student in our lab with myself and Richard Anderson, Kelsey Grieve, and she did this in collaboration with Oxford, Suzanne Williams. So when she gets their somatic cells and she places just mouse somatic cells under the kidney capsule, you see that after a period of time, 21 days, then it's, there's nothing, there's no germ cells, as you would expect. But when she combines these putative oogonial stem cells with somatic cells, what she finds is that there are follicles that have been formed. And so the follicles, the oocyte within these follicles appears to be more looks like a bovine and has more of a growth trajectory of bovine. And Kelsey's now doing detailed RNA analysis of these cells. But it does look like these cells can go down the pathway of germline development under the right conditions. So making these oocytes from any stem cell, be it an induced pluripotent stem cell, an embryonic stem cell, or a unipotent cell, such as the cells that we can isolate from the ovary, is a complicated process. We have to get them to the point where they would form primordial follicles, grow, go into meiotic arrest, acquire competence, acquired developmental competence, transcriptional repression, and genomic imprinting. So obviously, at each stage, this has to be tested to see if it's a normal progression of development. And again, that's the challenge ahead for us. But what does it mean if we found these cells, and we find them in adult ovaries, we find them in prepubertal ovaries, we find them in Turner syndrome ovary, and we find them in postmenopausal ovary? What does it mean? Well, the menopause happens. That's a fact. A lot of people say, well, why do you get the menopause if you have these cells? Well, that's another issue, another question. But cells that have germ and stem cell markers can be isolated from the ovaries of women and prepubertal girls. And this is a useful model for germ cell development in human. And that's what we're lacking, because we tend to focus on the mouse, and of course a mouse is a mouse and doesn't really tell us too much about human germ cell development. And of course there are potential clinical applications, so we have to do this basic work in order to see how far you could take this in terms of applying cell-based strategies for fertility treatment. So I would end by saying the field of germline stem cells is new. And a lot of the concentration of the effort has been in doubting their existence. And that's good. It brings up a healthy debate and so on. But there are so many papers now that are published showing negative results. But there are so many people that are also getting positive results. And so we have to have a balance and find out exactly what does this mean. So it's time now to really concentrate on defining these populations that have been identified in a range of species in bats, cows, pigs, rats, monkey and lemur. And they've also been found in the mouse by some groups, but not by others. And it's only by understanding these cells that we really can get to the point where you can apply cell-based strategies to treat infertility and fertility preservation. So I'll end by thanking the people who do the work. Maria McLaughlin, Yvonne Clarkson, Sheldon Locke and Kelsey Grieve have been the main people working on this project. And of course, clinical collaborators, Richard Anderson and Hamish Wallace, and the women who donate their ovarian tissue and of course give us consent through the Fertility and Preservation Programme as well, and the Medical Research Council for funding this work. And thank you very much for your attention.